So thanks. I'm going to briefly touch on some slides that Nico already showed, and I'll go through those relatively quickly. My disclosures have to do with having received research materials, and these are collaborations. The first I'll show you some data from, the second I won't. And so I think that we've covered a good deal about mesenchymal stem cells and why we should be interested in them. I'm not sure we completely know how they work mechanistically, but we've got some ideas. They were originally discovered in bone marrow, but can be sourced from a number of different places. They exist within the body around blood vessels, and so we say that all MSCs appear to be pericytes. And there are other sources such as adipose, gingival, and umbilical cord. And an umbilical cord, they actually live in the Wharton's jelly, not in the cord blood itself. This is the original, just from one of the original figures. It's an adaptation of mesenchymal stem or stromal cells, and they may not truly be stems, so Nico may be more correct when he actually calls them mesenchymal stromal cells. And the original hope was that these would repair bone and joints, and we'd stick them in, and you'd get articular cartilage, and everybody with OA would be cured. And that's really been an elusive goal, but it's been much clearer recently that they have tremendous immune modulating properties, and that might be their greatest advantage. They appear to home to sites of injury and inflammations to repair it. So if you have a scar or a burn or a joint, and they appear to tune down the mechanisms of the immune response, but they are hit and run, and they appear that, at least in an allogeneic fashion, that they induced host reparative mechanisms, and it's not just by their sticking around that these things occur. Nico already mentioned that they have low class 1 and class 2 MHC. So, for example, you can test your cells by putting human MSCs into mice. Mouse MSCs are not exactly the same as human ones, and you can give one person another person's MSCs without really expecting graft versus host disease, as they're largely invisible to the recipient system. So we became interested in the idea that seropositive patients with RA, over 90% require lifelong therapy with whatever therapy they're on. And so we became interested in whether or not this particularly refractory population, could we actually cure it, or could we reset the immune system? And I think the same, as I've learned more about the patients with systemic JIA who are refractory, I think the same questions get asked. There are also reports of catastrophic diseases like scleroderma and lupus nephritis, for which there appears to be at least anecdotal evidence that there might be evidence. Now, so we decided to go with RA both because of some of the source of our early funding, but also from a regulatory standpoint. I think, as Nico mentioned, it's easier to go into adults than children. And since I'm dually trained in pediatric and adult rheumatology, this was a target population for me as well. And so we've already talked about patients with RA not achieving sustained disease off therapy if they're seropositive. And for those juvenile arthritis patients who persist into adulthood, they appear to spend anywhere between about 40 and 60% of their time with some form of active disease. And so we wanted to know if these cells that we had could alleviate symptoms and halt damage. We know that these cells are expensive, but so is a lifetime of biological disease-modifying anti-rheumatic drugs. And so we developed a hypothesis that you would be able to do an immune reset and try and restore the immune system to a pre-RA or JIA possibility. I think that Nico has already been over these points. So we chose in the lab to use the TNF-alpha transgenic mouse, and this is a mouse that constitutively expresses TNF-alpha, so you can't shut it off. And so eventually all mice are going to get arthritis. But it was useful to show some localization of mesenchymal stem cells, and we tried intra-arterial injection, and we ended up killing the mice because, unbeknownst to most people, these mice have a bleeding disorder. And so if you try and go into the carotid, you're not likely to be very successful. And so we did do IV and intraperitoneal, and we were able to show localization at about 2 to 3 days into inflamed joints. And certainly the cells traffic to the lung, but we also think that they traffic to spleen and to resident lymph nodes. And our goal in using this model was not to cure arthritis, because we didn't think we were going to be able to cure this constitutive expression, but to slow down erosion. And so it's not a great model to induce Tregs in, whereas the model that Nico used, and I have a slide in here of Jost's paper and Nico's paper, but we thought that if we could get a hint of efficacy, that these cells had the capacity to reduce erosion, that we would have some basis to go forth with a trial in humans. And the possibility that MSCs can either induce regulatory dendritic cells or regulatory macrophages is also of interest to us. So what you're looking at is on the left is the TNF non-transgenic littermate, and so we sequentially gave these mice starting at about 6 weeks, 1 million MSCs every 2 weeks, and we did a series of micro CT scans on them. Now this is 6 to 8 weeks in a TNF transgenic mouse is before you see a lot of clinical evidence of arthritis, but if you sacrifice them and look on histology, you'll see evidence of inflammation. And so what you'll see in the top right is that there are clearly erosions in the TNF-alpha transgenic mouse at about 14 weeks that didn't get treated with MSCs. But I believe that you can see attenuation of erosion in the bottom right panel, and that the bottom right looks closer to the TNF non-transgenic than the untreated mouse. And so this was very encouraging to us in this very robust model that we had any sort of effect. These are the histology diagrams. This is alizarin red and saffron and O. And what you're looking for is deep red staining of the cartilage. So in the A panel, you can see that in a black 6 mice that doesn't have the transgene, you get nice staining of the joints. B is a non-transgenic littermate, and it's close to the A panel. However, in C that got PBS instead of any cells, you can see that there's fading of the red, and that there's a tremendous amount of infiltrate. And we also have serial sections that show H&E, and so this is a combination of both lymphocytes and some neutrophils. And then in the TNF-alpha transgenic MSC mouse, you can still see a little bit of inflammation, but this appears to be more preserved than this, which looks somewhat washed out, and the joint space appears to be preserved. And about the same time that we're doing those experiments, Nico's group in the Netherlands published this, and I think just to point out that this is really, this is a normal uneroded joint. This is a treated joint. This is an intact joint, and this is a joint that has, this and this are eroded joints here, and this is preservation of the joint space. So now I think you have your evidence that in 2 models that mesenchymal stem cells were able to affect the joints. So we knew they were well tolerated in mice, and that made up some of our preclinical data for the FDA. We thought the potential for efficacy in humans were good. We're interested in whether or not there's improvement in Treg number and function within our human studies. And we're thinking that the first target in JIA may be refractory SJIA. So our first study that we are in the process of getting up and running is a cell-based study to try and cure RA, or at least to reset the immune system. And we really needed a biomarker to understand early on whether or not these biologicals are having an effect, because it could take 24 to 48 weeks to see a clinical effect, and this is going to be a small trial. And so for the people who are not familiar with looking at this, what we're looking at is we're looking for T regulatory cells that live up here, and these are CD4, this is CD25 or IL-2 receptor, and the ultimate test is not what these cells have on their surface, but what they do functionally. And so we did an assay, and this is what we do is we load CD4 T effector cells with a vital dye, and we look for division. So each time the cell divides, there's half as half as much die in the next generation. So you can very nicely, doing a flow-based assay, look at proliferation. And so the first thing we showed is that our assay was working and that T regulatory cells, at least at a ratio of 1-to-1, both in healthy donors and RA patients, appear to suppress proliferation, which is what the literature says. We worked with this company called Athersys that has a product that doesn't yet have a labeled indication and their product is multipotent adult progenitor cells, which is slightly earlier than MSCs, and so they have more expansibility, and they're in trials for stroke right now. They're really ramping up their stroke program. And so what we're able to do is show that conditioned media basically stop proliferation. So you see suppression of all this stuff after culture with conditioned medium. And the same is true with cells from an RA patient. These cells were stimulated using a bead-based assay that had antibodies conjugated to CD2, CD3, and CD28. And so we're interested in using potentially their product, and we explored it, but as Christina alluded to, as a company, you have to focus, and you can't do everything at once. And so it became clear that we were really going to need to use our own shop, and Case Western has really been a leader in this. And so we repeated many of the experiments with MSC conditioned media made in our core, and we were basically essentially able to show suppression of proliferation. And so this was encouraging that we had a product that we could go. We talked a little bit already about the safety. We minimize infusional toxicities with Tylenol or acetaminophen, and no tumor formation, even though it's a theoretic risk more, I believe, of autologous cells than allogeneic, has been observed. There are companies, and I won't go through all the data in the interest of time. This is from Tygenix that used an adipose-derived cell, and I assume one of your PIPs is from Tygenix and the other is from Mesoblast. But they were able to, and they used biologic refractory patients, and that's very important. So we know that biological refractory patients are more tough to treat. But they did show some trend when they advertised everybody, when they looked at all their groups, that with the cell product, that there was some trend towards having an ACR 20, 50, and 70, and even some patients appear to achieve DAS remission, so disease activity score remission. Mesoblast has done a press release. The data is not available in manuscript format yet, and they also reported improvements in biological refractory RA, and I think this is a trial to watch. Again, just to point out dosing, they used 1 million per 2 million per kilo, and this is a little different than the trial that we're going to go after. And so we have a target of early rheumatoid arthritis, and this is actually the first trial I know of that's letting people do MSCs prior to biologicals. Patients will, as is required by the FDA, remain on methotrexate. We're allowed to hiatus it from 3 days before to 1 week after. And there is some literature from our hematologists that methotrexate is not going to be too toxic to the cells. It's a dose-ranging study, so we'll use 2, 4, and 6 per kilo, and we'll go up and we'll look for dose-limiting toxicities. We've got an IND. We've got the IRB approval at one of the sites, and the IRB approval at the second site will probably be forthcoming this week or next week, and it's been funded by NIAMS. And so I think the issues as we go forward in looking at systemic onset juvenile arthritis is what's the best clinical design. We didn't originally design our RA trial with placebos, and we were asked both by the study section and by the FDA to consider the use of placebo patients because it at least makes biomarker data available. I think this is really important, at least on this side of the ocean, which is we were told in no uncertain terms that if we were to go into phase 3, we would need to have a potency assay for the cells that were given. And we need a biomarker early on so that we can not do something if the cells are ineffective. So this whole idea of pre-qualifying cells or customization and licensing, I think it's probably wishful thinking to assume that the same cells in RA might be the same cells that might work in lupus or might work in systemic onset juvenile arthritis, and we need to define what the biomarker outcome is that we're looking for. If there's a need to use a placebo in this trial rather than do it open label, some thoughts I've had is, could we do it phased? So in the RA trial, if we see changes at 1 week in the biomarkers we're looking at, could we do 2 infusions, 1 saline, 1 cells at week 1 and 2 and sort it out, or week 1 and 3, and would that end up being an acceptable kind of trial design? So the process for development going forward, there's been an application submitted to the Arthritis Foundation that they're very interested in. We're not, we don't have a notice of award yet, but we're moving in that direction, we hope. There needs to be protocol development, and some of you very graciously got up at an ungodly hour this morning to do a 7:30 a.m. focus group with us. So we'll do focus groups with parents, physicians, as well as some teenage and young adult patients who've had a disease like this, or some may end up being really refractory polyarticular kids also, but we'll attempt to get kids who've had systemic onset. Discussion of steroid taper has yet to occur, because I think that's an important issue, as it wasn't a rapport in any other studies. And some of the regulatory requirements that we encountered but didn't necessarily anticipate is the need to send out in the current environment what's called adventitial testing for extensive viral testing. And so this was a $10,000 to $12,000 budget item that we didn't foresee at the time that the grant was initially submitted. So we've identified 6 sites, budgeted for 12 patients, but we need to demonstrate safety, and it would certainly be helpful to have some indication of a biological effect prior to proceeding to a phase 2 study. Advantages, I think, have already been highlighted. Infection's relatively minor in terms of the concerns. It's expensive, and currently it would be difficult to produce that amount of cells on the same scale as a DMARD or a biological DMARD. None of this work gets done in isolation, and I really owe thanks, especially this weekend, to Sue LaSalvia and Stephanie Knut, as well as the SJIA Foundation for sponsoring this, and all the people who participated this morning and who are going to be sites in this work as we get it off the ground. So I'll stop there. Thanks.
So you showed the key point of the effect of supernatants on cell proliferation. So as if soluble factors or single soluble factor could explain part of the whole effect. Can you tell us more on the possible options?
So I think there are both, I think in vivo that there are contact dependent and contact independent things. I think that for RA, the capacity of mesenchymal stem cells to secrete IDO, which is the rate-limiting step in tryptophan metabolism, production of PGE2, and those kinds of things are going to be important. Our plan for the RA prequalification of cells is actually to measure IDO and PGE2, as well as look at proliferation of T effectors or suppression of proliferation with soluble products from the cells. And so we will compare different cell donors. The other point that I did want to raise in respect to CMV is that we've chosen, as other trials at our institution have chosen, is we're looking for CMV negative donors to try and reduce any impact of the potential for viral infection.
Just to add, because we are looking at this question with a different idea in mind, which is migration of cells to the inflammatory site, where they will interact with the local mesenchymal cells. So we use a number of bone marrow, synoviocyte, or whatever, and our conclusion is slightly different in the sense that when you have migration of activated, and the key point is activated cells in contact then with mesenchymal cells, more or less you induce inflammation, and back to IL-17 as an example, you induce massive secretion of IL-17. So my understanding is that you will inject cells systemically. In the mouse it was local or IP, right? And from there, you can't demonstrate the migration of cells, possibly because you don't inject enough to follow them. But the point could be that when you inject them, they will obviously go somewhere, and from there, through local interaction different from the inflammatory site or not, secrete regulatory factors, immunosuppressive or whatever, which will then act systemically, leading to the clinical effect you see. Is that the understanding you have?
We also saw a reaction in contralateral.
Right, yeah. Because there have been also results, slightly different idea, but which is the pathogenicity of the mesenchymal cells, the synoviocytes, right? And in the skid mouse models, injecting on one side
right.
And
that's the Smolin paper. That's the paper, I think, from
Nature
Vienna.
Medicine,
from Vienna, and that's so.
And seeing the cells migrating to the other side, right?
So I also wonder, anyway. In your experiments, are you using mouse mesenchymal stem cells or human?
I'm famous to try to cure human disease, but
because mouse mesenchymal stem cells can be different, so
sure.
So it would be.
It's clear also that cell-cell interactions are critical, and step by step we are identifying molecules which are critical for this local and possibly systemic inflammation. We could at the end, or will be at the end, the key targets. Right?
Okay. Did I answer the question?
No, but yes and no. Yeah, of course, it's part of it. But because out of these sort of immunosuppressive factors, one is TGF-beta, right? Because you mentioned a lot regulatory cells, and the other one, not so clear, could be the IL-10. So back to biomarkers, this could be or should be somewhere on the list, right?
Agreed.
Okay. Questions?
So I just thought it was striking that both of the talks that we heard, the researchers decided to end up going with making their own MSCs rather than going with a vendor. And I'm wondering if we should be looping these stem cell vendors into future conversations, how that decision was made, just discussing that topic a little bit. Because, I mean, if somebody's already going through the phase 2 process, getting to the phase 3 process, then that's an interesting thing to this community, right?
Yeah. So I think that I've not talked to Tygenix. My collaborators have some conversations with Mesoblast, not about this particular RA, but more in general, and maybe early on. And I think that since it was RA, that maybe a company's not necessarily interested in giving their cells to us, but potentially it could be a conversation in the JIA indication. Nico, what do you think?
Yeah, technically, certainly. It's also a matter of costs. But this could be discussed, because if companies could do it much cheaper than we can, and it's expensive, all the laboratory work.
So if we could satisfy their PIP, then maybe they'd be more eager.
What about Aline Boutencourt? Yeah, like she had a startup in this domain. Aline.
Yeah, I did.
And she has a startup, and I think right now she's targeting something else. But if you take a smaller company which is interested in developing a product in this domain, and you work with them, maybe it'll be a mutually beneficial thing.
So early on, I will say for phase 1, that when companies do a phase 1, I think that the general feeling is the regulatory burden overall is higher than when it's an academic investigator's IND. And so my experience has been that while the FDA wanted certain things, that they've been incredibly helpful. And I think it's more, it may be easier for an investigator doing a small study, but the cost of the regulatory burden when it's a company rises. I don't know if you can comment on that at all.
Well, if you speak about money, pediatric investigation plan submission is free of charge. Also, scientific advice for pediatrics is free of charge. Orphan designation gives some incentives for decreasing the fees for marketing authorization applications. So I don't think it is about money as for regulatory decisions.
It's maybe that the reason that why most academic centers do it themselves is that it's so very, very simple to isolate and obtain the MSCs. Characterization is another ballgame because we know they're heterogeneous, and these companies will also have this problem. I think.
And so cells cultured in Arnie Kaplan's lab versus the core, where they're not cultured with serum, they may look different and have different effects.
Sure. Yeah, that was going to be my follow-up question. Can we sort of, because it's almost like there's a variety of different dimensions upon which MSCs can vary, right? And then there's also the question of dosing, right? So it's an n-dimensional space, and if this is a potential treatment for this disease, it doesn't necessarily mean that the MSCs that you made, or the MSCs that you made, or the MSCs that Company 1 made, or the MSCs that Company 2 made are necessarily going to be the key, right? Like, so you kind of you actually probably want some variety in terms of the approaches.
We do test for the trilinear differentiation and suppressive capacities.
Of course, yes.
But harmonizing.
But so, like, is it from bone marrow? Is it from lipid? Is it from blood?
But whether, for example, we could actually harmonize manufacturing processes so they looked across the same at both sides of the ocean, I think is something that will.
Well, do you want to do that, or is variety better? Like, if you do it different and one works and one doesn't, then if you'd done it both the same, maybe you both would have done the one that didn't. Yeah. So it's so I guess there's just a lot of details within the individual lab. Like, you can't really characterize the number of dimensions because there's essentially a very large number. Is that the answer? Yeah. Interesting.
I think at least knowing what the differences are that we're doing would probably be helpful.
Yeah, and let's face it, it's an initial study, and of course we are very glad that we see some effects, so that will make us study more. But it's also, we have this grant, it's expensive, right? And we almost spend it, so we really have to think how to proceed with it. But then maybe companies can help. But if they are willing in this early phase to do that. Yeah.
So before companies have a labeled product, again, many of them are risk averse to having an investigator-initiated IND that could potentially tank their product.
Got it. Okay, so that seems like it's a core element of the dynamic. Makes sense.
Did you say?
That seems like a core element of the dynamic. That's essentially, the biotech companies are they're wanting to mitigate the risk and do in-house stuff so that they can control what goes on. Okay.
Yeah. Okay, I think we're touching on lunch, right? Is that correct?